Synthesis and characterization of iron oxide nanoparticles as MRI contrast agent for targeting and imaging of cancer. The blurring of boundaries between the disciplines of engineering and biological sciences has revolutionized medicine. Recent advances in domains such as nanotechnology have added many tools in studying biomedical mechanisms and added many weapons for the treatment of diseases. Iron oxide nanoparticles have been growing excessive importance because of their magnetic characteristic. Iron oxide nanoparticles, which are prepared, were employed to study their potential application as MRI contrast agent. PEG is to be coated onto the surface of the prepared nanoparticles to enhance the stability and biocompatible properties. The material, which is characterized and evaluated for the purpose of cancer cells, HeLa, targeting and imaging. Iron was measured using UV spectroscopy. The size distribution of the obtained nanoparticle is examined using scanning electron microscopy. Inductively coupled plasma mass spectroscopy is done for elemental determinations. Moving on to our aim is to synthesize and characterize iron oxide nanoparticle as MRI contrast agent for cancer targeting and imaging. Our objectives are isolation of iron oxide nanoparticles from bovine blood and encapsulation of polymer over iron oxide nanoparticle, characterization of the prepared composite material, in vitro studies using cancer cell lines, and fluorescence or MRI imaging. Moving on to the novelty, for the nanoparticles to be biocompatible, the isolation technique is done from the blood collected from the animals. This is one kind of extracting useful material from environmental waste source. The encapsulated and characterized iron oxide nanoparticles are targeted to image the cancer cell in the in vitro studies. As we all know, we know the structure of hemoglobin molecule, which consists of alpha chain, beta chain and the iron embedded between these alpha chain and the beta chain. It is not as easy as it sounds. It's not because the iron particles are tightly bound to the alpha chain and the beta chain, which is surrounded by 4 nitrogen atoms. Our work plan is as follows. We completed our synthesis work in the month of January, characterization in the month of February, and the in vitro studies in the March first week. Synthesis of iron oxide particles from bovine blood, which is considered to be the major part of our project. This synthesis process we have divided into 3 major divisions, which includes isolation of RBC, cell wash, isolation of heme. Isolation of RBC includes the centrifuge of the collected fresh blood at a required RPM and then to remove the supernatant. Moving on to the next step, which is the cell wash. We are adding a buffer which removes all the unwanted globulins from the centrifuged resultant blood. This buffer added blood is again centrifuged and the supernatant is removed. The last step, which is the isolation of heme, which includes 2 divisions such as acidification process and the neutralization process. The acidification process is done through the hydrochloric acid. It was tried under various concentrations. Then under a perfect concentration, the acidification percentage of HCl was fixed. And then to neutralize the acid added, we are using NaOH, sodium hydroxide. And the resultant powder was air dried. Material was air dried, and the resultant powder was gone. The PEG was coated onto the sample and given for characterization and in vitro studies. These images show the PEG coated sample. And the next part of a project is the characterization of the sample and the results. The UV spectra shows the relationship between the wavelength and the absorbance. The peak is said to be formed at 404 nanometer, which is our expected range. The ICP-MS result, which is inductively coupled mass spectroscopy, shows the presence of iron in the sample as 64 milligrams per kg. This indicates the presence of iron in our sample. Then moving on to the SEM analysis. The SEM analysis of the sample shows the presence of particles in 110 to 340 nanometer range. It was under our expected and desired range. And then moving on to the anti-cancer effect of the sample on HeLa cell line is shown as an MTT assay. The concentration it is found in the graph at 62.5. It was fixed as optimum range. The anti-cancer effect of sample on HeLa cell line is shown here. First, in the first image, a normal HeLa cell line is taken and it is treated with various concentration of the sample or the material added to the HeLa cell line. It was treated under various percentage or various ranges of the sample. The toxicity was checked at 1000 micrograms per ml, 125 micrograms per ml, 62.5 micrograms per ml and 31.2 micrograms per ml. 62.5 was considered as the optimum range. It could be seen that it has removed or destroyed mostly half of the cancer cell lines. This optimum desired range, which is 62.5, is then given for the fluorescence imaging of the sample. It conveys that the material can be used as a constant agent. This is our final expected result. At last, I would like to conclude that the work proposed novel techniques on isolation of nanoparticles from animal blood. PEG was coded to avoid aggregation of particles, that is to enhance the stability and biocompatibility. The PEG would help in avoiding the agglomeration of the particles. The sample was then characterized by UV, ICP-MS, SEM, etc. The prepared sample was tested for its efficacy against cancer cells. HeLa cells were cultured and toxicology studies showed decreased cell viability. The particles were imaged for its fluorescent nature and the result conveys that the material can be used as an MRI contrast agent. Thank you.